Thanks, Prashant, for the introduction and to everyone for uh, showing up here virtually, and excited to have a chance to tell you about some of the things we're the most excited about right now. And this will be a blend of, I think, some things that are recently published, as well as some stuff towards the latter half that is quite new and still not published or pre-printed. So, okay, so I'll just dive in here, right? So biology is full of trees, right? We have trees of evolution, as outlined by Darwin and species. We have trees of individuals, exemplified by Mendelian pedigrees. And of course, each of us ourselves is a tree, right? A tree of development of cells dividing and dividing again, right? We each developed from a single cell zygote, now, trillions of cells as a newborn, and many more as an adult. And the same is the case for pretty much every form of multicellular life on Earth, right? And you can, increasingly think about the genome as a blueprint for really specifying form and function in this program of development, which is remarkably robust, but also remarkably capable of giving rise to such beautiful diversity, as Darwin put it, endless forms most beautiful. So even though we have genomes increasingly for all of these species, there really only remains one organism for which we have a complete map of this process of cell division, by which the single cell zygote goes on to give rise to form and function so robustly. And that is, of course, the roundworm C. elegans. And so this, as the story is well known, I hope, to all of you, was deciphered by Sulston, sitting at a microscope, recently developed Nomarski microscope, for several years, to the point where the location on the floor under the chair in which he sat has like a big dent in it, apparently, if you go there. And just by marking by pen and paper what he saw under the microscope, he was able to completely decipher this lineage. And of course, he's taking advantage of the optical transparency of the worm, as well as the fact that its development is so stereotyped, occurring exactly the same way every time. So you could integrate observations made over many individuals into a single description, which is now 40 years ago. So here we are 40 years later. This remains the only organism for which we have this kind of a map. But this challenge is still sitting out there, and it's a great technology development challenge, which is how can one think about developing similarly comprehensive maps for more complex model organisms or other organisms, including, for example, the mouse, right? So the challenges here are pretty obvious. Mouse development is not, mice don't develop in a transparent way, right? We can't see into the womb, so to speak. Um, uh, Mouse development, like most multicellular organisms, is not invariant, meaning you can have a genetically identical mice and they will develop, the lineages will not be identical, and yet you will give rise to it will give rise to similar phenotypes at the coarseness at which twins are similar. And of course, there's the challenge of scale, right? So if we take the adult mouse versus the adult worm, we've got a factor of about 10 million here, right? Okay, nonetheless, let's try. And why? Why should we try, right? So in my head, I think about this as the phenotypic equivalent of the Genome Project. Again, the genome primarily being a blueprint for development. Um, one of the so I mean, we alluded earlier to, like, I have hopscotched around in various areas over the course of my career, but in the last couple of years we've really tried to hone in on development. And in kind of reading the literature during the pandemic, which is what I tried to do, really read textbooks, you realize, like, although an incredible amount is known over the course of many decades, it's not terribly systematized in the way that genomics has approached, the genome, for example, through the systemization of genes and things like that, right? So I think even though we know an incredible amount about development, there is still a value or a need there for the systematization of that knowledge. Development is a bounded process, and yet it includes everything. It's reproducible. A B6 mouse will develop the same way, and reproducible and accessible. A B6 mouse will develop the same way, in multiple individuals of a litter, as well as, 30 years ago and 30 years from now. And it is unquestionably an excellent model for human biology. Okay, so what we're aiming for here as we're thinking about this, it's not going to be a perfect lineage. It's also not going to be just a big list of cell types, right? So we think that there's something in between that kind of captures the flavor of typical multicellular development. And we've tried to frame this as we call this a consensus ontogeny, where you think about essentially like a tree of development, right? Where there are aspects that are invariant from individual to individual, and there are aspects that may vary even between genetically identical individuals. And there's still a, kind of a conceptual and practical challenge of how to reduce this incredible complexity to something that we can get our heads around and yet captures the richness of the process. And that's an ongoing challenge. Okay, so now stepping way back, what are kind of the major avenues of attack that we might take on this goal, right, of trying to reconstruct mammalian development? So, as mentioned, I broadly think there are kind of three strategies one could take. So the first one is the approach that Sulston took, which is to simply look, right? And there's some work from Kate McDowell and others showing that you can actually peer in and see the developing embryo for certain windows, or you can try to culture it ex utero. But the complexity and scale are such that we're probably not going to get a Sulston-like lineage simply by looking, right? So let's take that a bit off the table. So I'll talk about the second strategy very briefly to give a sense of what we've done and why we think there are limits. And then I'll come to this third strategy. So the second strategy I'll call sampling. And it's a pretty basic idea, right? So this is genomics, where you destroy everything and you at one moment of time, and you try to measure everything in that sample. And so we can, if we're looking at a mouse embryo at a particular moment of development, we can do a very good job of characterizing that moment, but we miss out on what comes after and what came before. And so we can address that to some degree simply by doing a lot of sampling, right? And so of different embryos, and then trying to piece it all together after the fact. And so this is what we did, focusing on everything from mid-gastrulation, so about day 8 in mouse development, all the way out to birth. And working with Jackson Labs and Steve Murray's lab in particular, we tried to get as many snapshots as we could, and then to profile them with single-cell RNA-seq, right? And so, I think with the maturation of not only multiplex sequencing technologies like next-gen sequencing, but also other modalities for improving scale. This is the kind of project that I think 20 years ago would have been only the province of a large-scale center, but the work I'll briefly talk about was really just done by 3 people, right? So Ian Murray sorry, oh my gosh, I have a typo there. Ian Welsh at Jax. I combined the PI's name with the postdoc's name. Ian Welsh essentially staged 500 embryos that he isolated from mouse embryos, and picked 74 of those that he felt kind of represented intervals that we would then sample, right? So for early development, we can count somites, and so we can get what corresponds to 2-hour resolution. And then later in development, Ian relied on both automated and kind of more subjective aspects of what he saw to estimate the timing of those embryos at about 6-hour resolution. And then later, it was really just kind of knowledge about what embryos look like at various stages that allowed him to line those up. So we've got 2 hours resolution at the very beginning, and then about 6 hours resolution all the way out to the rest. In order to enable single cell profiling at scale, we rely on a class of methods that our group, together with Cole Trapnell's group, developed some years ago called combinatorial indexing, or sometimes split pool sequencing. And here we're pooling and, splitting and then pooling populations of nuclei or cells in order to enable them to be combinatorially labeled in a way that essentially allows us to profile vast numbers of cells or nuclei without ever really isolating a single cell or nuclei. And I won't talk about these in detail because I think the methods have been out for quite some time now and I think are appreciated. But they're matured to a point where Beth Martin, who's a research scientist in my lab, could, with her own hands, over the course of about 6 weeks or so, generate a 10 million plus single nucleus RNA-seq data set that essentially covers those 74 embryos spanning all of those time points, right? So a big data set. And then, the last piece here is the analysis. And here also, I think the tools have matured just so remarkably, I think, enabled in large part by open sourcing and all of these other things. But Cheng Sheng Q also goes by CX, was able to take all of this data and wrangle all of it and really also get his head around mouse development, annotating hundreds of cell types and kind of piecing it all together, essentially resulting in kind of a first draft map of mouse development that, even though we started at gastrulation, we leveraged some other data sets that went further back and we're able to kind of put together a V 1.0 roadmap of cell types that starts at the single cell zygote and goes all the way out to the cell types present in the newborn pup, right? So an extremely reductionist view of development, but kind of a scaffold that we can build on. So there's a lot more in the work here that I won't have time to get into. The paper was published last year. If you're interested in any of those deep dives or any other aspects of how we did this, but I want to make the broad point that this is, I started with the example of Selston, and what we have so far is kind of the Cliff Notes version of this, right? Where we have a map of the cell types or like an abstract painting kind of thing. We roughly know what's going on, but everything is inference between between different mice, and we don't really have the beautiful map of every cell division and a precise delineation of exactly what happened in exactly one mouse as its development unfolded, which was really the goal here, right? So that brings us to the third paradigm in our list, which is this concept of recording, right? Okay. So so the broad idea here, right, is to ask whether we can turn cells and organisms into their own historians. To put it another way, can we program or add additional functionality to the genome that allows cells to essentially capture aspects of what is going on and to record those observations, so to speak, to the genome itself? So we are going to use the genome both to encode the machinery to do recording, and we are also going to write to the genome. We're going to write those records to the genome itself so they can be read out subsequently, right? So this is like this is a thread that goes way back in my own trajectory, right? This was actually my very first project in graduate school with George Church and Susan Demecki, who was one of the people who pioneered the use of recombinases in mice. And the basic idea was to build, this was before things like Brainbow. We were going to build a giant array of flipping bits, and somehow those would just, those zeros and ones would just, flip and flip again. And that information, if we put that system into a mouse, would at least on paper, would provide enough information that we could do lineage reconstruction. And this got us, we only got a few months in here. It only resulted in the patent application, which was never issued, and nothing ever came of it. In part, well, I'm not sure anything would have come of it in that time frame, but it's one of those projects that I think was maybe ahead of its time. And to my luck, a postdoc tracked me down after my first lab meeting and suggested that I change projects. And I thankfully listened to him, because I think it would have been a very long PhD if I hadn't, and switched to other things. But it kind of stuck in my head. And many years later, came back to this with my own lab after the emergence of CRISPR, pioneered by Prashant and others in the early 2010s. So the basic idea, and this was an idea that many groups had around the same time, was to leverage CRISPR-Cas9 in order to stochastically and irreversibly mark lineages during the course of biological development. And so, in a little more granularity, imagine that we engineer into a zygote, which we did, a zebrafish zygote, an array of CRISPR targets. And we also, we inject Cas9 and we inject guide RNAs into that single cell zygote as well. And then as the embryo is developing, cells are dividing, you've got double-stranded breaks that occur in this array, followed by NHEJ, non-homologous end joining, which gives rise to a variety, a diversity of mutagenic events, including insertions and deletions. And in any given lineage, these edits will accumulate, but a different set of edits might accumulate in some other lineage. And we measure this at the end. And then these are every line here is what we see in a single cell. And you can see patterns that are shared and patterns that are unique. And we can rely on kind of standard phylogenetic methods to reconstruct trees based on this information, where the trees are reflective of cell lineage, right? And so this works on paper. It actually worked in a fish, right? And so the future was bright, but this is now some 8-9 years ago. And part of the reason I think for the gap that we and others had challenged really taking this to the next level was that the devil's in the details, and double-stranded breaks are terrible for cells, and can do a lot of bad things, right? So on the previous slide, I showed some kind of nice looking data. This is other data on the top right here that unfortunately is also real data. And this is our array of CRISPR target sites, and the red bars correspond to deletions. And you can see that it's a bit of a mess, right? When you have multiple double-stranded breaks occurring within a tight window, it can lead to deletions, which would delete information, delete our recording capacity, etc., right? And this makes things like lineage inference very challenging. So that's one big class of problems. It's just the nature of double-stranded breaks are such that we could do recording, but our recording destroyed our recorder, right? And also destroyed our information. The other big challenge, illustrated on the bottom right here. So Alex Shear, other groups went on to take Gestalt and show that you could combine it with single-cell RNA-seq technologies, which were emerging around the same time. And we could build these lineage trees where we knew not only the tree, but we also knew the cell types of the terminal nodes, right? So we read out, the final states by single-cell RNA-seq. We also capture lineage information. We build our tree. And so that's cool. But what this lacks is that we can infer that certain ancestors existed, but we don't really know who they were. And we don't know what happened along the way, right? And this contrasts with Selston, who could actually tell you who the ancestor of any particular cell was and where it was and all of that, right? Moreover, the other challenge here is that we're not really recording anything here other than lineage, right? And so what we really would like to do is to know everything that's going on inside of a cell, not solely how the cells are related to one another. Okay, so a couple of years ago, as we continue to percolate on these ideas, Junhong Choi, who was a postdoc in the lab, now has his own lab at Memorial Sloan Kettering, developed this concept of DNA typewriter. And so here, the analogy, if you think about the previous technology I talked about, which we call Gestalt, is like, shooting a paint gun at a wall. Typewriter is much more elegant and much more analogous to a typewriter, right? So in the sense that we have a single type head and we're punching keys. And every time we punch a key, the type head moves one position down on our recording media, right? So it's a good name because it's the analogy actually holds pretty well. And so in molecular terms, how do we actually do this? So now we're relying not on conventional genome editing, but rather on prime editing, which was developed by David Liu and colleagues a few years ago. And prime editing relies on the fusion of a Cas9 nickase and a reverse transcriptase. And so that's one difference. And then the location to which we're writing, we call DNA tape. And so this is an exemplar of DNA tape at the top. And going left to right here, we've got a repeating 14 base pair monomer. And these sequences are designed such that the first 14 base pair monomer plus the first half of the next 14 base pair monomer plus this key sequence at the beginning, so those 3 bases, all together forms the target site for a guide RNA. And the prime editing guide RNA or PEG RNA is encoded not only to introduce the nick here, but also to write an insertion. And that insertion has 2 components. One component is a barcode, which is encoding information. The other component is the key sequence. And so in the course of an edit, we write information in the form of the barcode, we inactivate the type head, and we kind of create a new type head 1 unit down. So effectively, we move the type head 1 unit along the tape. And this process can be repeated, with subsequent monomers, right? Okay. So that's the idea. Does this work? It's one of these things that works on paper. You try it, it worked the first time. We were able to show that if we have multiple dinucleotide edits that are being programmed to a tape, we only get editing in kind of this directional fashion. If we see an edit at the 3rd monomer, it is always preceded or almost always preceded by edits at the 2nd and 1st monomer and that kind of a thing. So that's good. We did some fun toy experiments where we, for example, tried to write text messages to ourselves in cells and then recover that information. So here, for example, you could imagine encoding different letters of the alphabet as different trimers, writing those through a series of transfections in order, and then trying to recover that information. And we can get, approximate reconstructions of what we intended to encode. Here's some more examples. So Mr. Watson, come here. What hath God wrought? We even tried a K-pop lyric. And in each case, we're able to get something that was pretty close to what we tried to encode. All right, so those are fun experiments. Now, the more interesting experiment is to try lineage. All right, and so here we start with a single HEC293 T cell into which we've engineered lots of tape, many PEG RNAs that are writing different barcodes to that tape. And we got lots of these tapes, and we also have the prime editor as well. We expand this, and we presume that we're going to accumulate these kinds of ordered edits to our tape. And then we recover the cells at the end, do single cell RNA-seq. And so this is kind of representative data. So every line here is 1 cell. And we have lots of tapes. So every set of 5 here is a different tape. And you can see that within each set of five, we are getting this left to right writing. And in some cases, we've completely used up the tape. In other cases, there's still blank spaces at the end at the time we terminate the experiment. And then you can see shared patterns. And on the basis of those shared patterns, we can reconstruct a tree, right? But now we're not dealing with deletions and all of that business, right? And so we're really limited by our scalability of our single cell technology. So now here, this is a tree that's much larger than the Celsin tree, but it's also much more boring. This is just expanding HEC293 T cells. But the point is that in a single experiment, we can build these large trees in a way that's the scalability is just up to how much single cell RNA-seq we can do. Okay, so to kind of tie this together, at this point, we've got DNA typewriter solving nearly all of our woes, but not all of them, right? And so we've got ordered recording. We are not destroying our media. We can write continuously over several weeks. In principle, we have many symbols. Every insertion is a different symbol. So if we have 2 base pair insertions, we can encode 16 things. If we have 4 base pair insertions, we can encode 4 to the 4th things, so on and so forth. We've avoided double-strand breaks, and there are other advantages here. But it's still boring, right? So we've got it's boring at least till the end, right? It's a little bit, the analogy here is like a monkey at a typewriter, right? So if we have a monkey at a typewriter inside of our founding cell, and it's just punching keys on the typewriter, and that cell divides and passes on what's been typed so far, together with the daughter monkeys, we can use that to reconstruct a tree, but we don't know what happened along the way. So how do we impart meaning to particular keys? Biological meaning. So this is a project that was pursued in parallel by Choi in collaboration with Will Chen, who was a graduate student in the lab at the time. And the challenge here is that in principle, we could have a specific insertion for every signal of interest. So if we're interested in a particular signal, we could simply have that PEG RNA be conditional on that signal being present, the PEG RNA writing that insertion. The challenge here is that although there are many enhancers for different kinds of biological signals that we'd be interested in, which would lead to great reporters, those tend to be Pol II promoters, whereas conventionally people drive guide RNAs from Pol III promoters in mammalian systems. So Will came up with this approach where we find a way to essentially have the levels of, or the production of particular PEG RNAs be dependent on the activity of a Pol II promoter, which is in turn, driven by a signal-specific enhancer. So we've got an enhancer driving this Pol II transcript, which encodes a endoribonuclease, which then goes and cleaves these hairpins, releasing the guide RNA to write a signal-specific insertion. And so this turns out to work really well after some optimization. And so these are dose response curves where we have particular agonists for particular signals, and the y-axis represents, not response in the classical, like turning on a signal, but rather the amount of editing that we're seeing with a particular concentration of an agonist for this pathway. And the cool thing is that we can multiplex this, right? So we can have many of these recorders present in the same system, with each enhancer driving the production of a PEG RNA encoding a different insertion. And so, if we've got, in principle, if we've got 5 base pair barcodes, we can encode 4 to the 5th or 1024 potential signals, right? And very briefly, I think for reasons of time, I'll just mention this really quickly, that we can combine typewriter and this newer system, which we call Engram. And we have shown that we can, for example, record the order of 2 signals relative to one another, and we can record more complex patterns with which these signals are applied. This is a pretty early area, and I think there are huge opportunities for doing things that are more sophisticated than what we've done so far. But at least in principle, the combination of DNA typewriter and Engram can work. And so we're super excited about that. Okay, and I'll skip this slide just for reasons of time. But at the highest level, I think what we have in kind of um, in high level terms is the components for a symbolic language for longitudinally recording biology, right? So we have our tape that we're writing to. We can have a multitude of different symbols being written to that tape. Some of those symbols can be analogous to a monkey at a typewriter, keys that are getting pushed without thought. Others of those symbols can be driven in ways that are conditional on biological signals. And if this all works together, and things appear to be headed towards that, we have the possibility of generating, this is a phrase that Michael Elowitz termed decorated trees, right? So lineage trees that include additional information layered onto them that tell us not only how cells are related, but also kind of what happened along the way, which is what we were interested in. But everything I've told you about so far is still in HEC293 T cells, right? And so what we really want is to do this in a mouse, right? And so we're not there yet, but we're marching in that direction. But I'll kind of tell you about steps that were positive steps in that direction. And I'll do that through the lens of a different system, which I'll get to in a second here, right? So I started out early off with this slide, right? So endless forms most beautiful. All of these species exist in nature. And everything starts as a single cell zygote, right? In the lab, particularly in the past decade, we have this panoply of stem cell models, right? Organoids, gastruloids, all kinds of things, right? And these are also beautiful and also seem to have endless forms. In this case, for the most part, with some exceptions, the starting material is not a single cell zygote. It's an aggregate of stem cells, right? Of a few hundred stem cells that are the starting material for making one of these organoids or stem cell models. Okay. So endless forms most beautiful can also be endless forms most irreproducible, right? So it is like it's not as much in the literature as one might hope, but if you're into this field, it's inevitably the case that we see things like this where even under identical conditions, batch condition, the same batch, aggregates of stem cells can give rise to lots of different forms, right? And the reasons for this are still not entirely clear, right? So we have set out to investigate this partly as an excuse for moving our systems out of HEC293 T cells into stem cells. And in particular, we've been focused on gastruloids. So these are a powerful model of early development pioneered by the Arias lab. And they kind of, effectively model the posterior mammalian embryo. We've got an aggregate of stem cells that is exposed to a Wnt pulse, Wnt agonist pulse, resulting in symmetry breaking, primitive streak formation, and kind of a tail bud morphology. And kind of the main thing going on here is the induction of neuromesodermal progenitors, which are a cell type that will effectively give rise to both the mesoderm and the neural cells of the trunk, right? So thus neuromesodermal stem cells or progenitors. And you can see here at the bottom, individual gastruloids still exhibit a lot of heterogeneity, both morphologically on the bottom. This is not our data. This is from Prisca Liberali. And then if you look at the cell type compositions of individual gastruloids, we also see quite a bit of variation. And this is a reanalysis of data from John Marioni's group where they pointed this out as well. Okay, so what is the basis for this heterogeneity? So I'll call it intra-batch or inter-individual heterogeneity, right? So after you seed the aggregate in a well, you can imagine that there are microenvironmental differences or stochastic things that contribute. But we first wanted to focus on the fact that we're starting with a polyclonal aggregate, right? And so there might be variation between the polyclonal aggregate in one well and the polyclonal aggregate in the next well. And so Sam Regalado, who's an MD PhD student in the lab, tried to address this by developing a system whereby we could instead start with a monoclonal aggregate, right? And so what I mean by this is that Sam devised a way to start with a single cell, seed that, and grow it up on a layer of feeder cells to an aggregate that he then lifts over to a new well for kind of the more standard induction of a gastruloid, right? So we're still starting with an aggregate of stem cells as we induce the gastruloid, but these cells are now as related as possible, right? They really started from a single cell just a handful of generations before, right? And these monoclonal gastruloids morphologically develop similar to conventional gastruloids, conventional polyclonal gastruloids. So interesting question, are these things still heterogeneous? And the answer is they are. They're actually arguably even more heterogeneous. So we can do this in plate format. So now we have over 100 of these monoclonal gastruloids. We can tag these, we can put sequence tags in or barcodes into these founder stem cells. And then we can do single cell RNA-seq on a whole bunch of monoclonal gastruloids. And then we can actually figure out what cells came from what monoclonal gastruloid by also sequencing the debris left over in the well from which we harvested a particular monoclonal gastruloid. So we can group the cells together as having come from the same gastruloid, if that makes sense. And we see lots of cell type diversity still. We actually see some cell types that we didn't see in polyclonal gastruloids. And then when we look at the individual at an individual level, we can roughly group these into 4 sets, right? So there are in the center 2 columns here are exemplars of the kinds of gastruloids that we expected, right? So we've got elongation in both of these center columns. And if you look at the cell type compositions of these gastruloids, they tend to be either mesodermally or neurally biased, right? So spinal cord, neurally biased or mesodermally biased. But then we also have a category of gastruloids that kind of fail to launch. They don't look like gastruloids. They're dominated by stem cell, like by things that look like stem cells, and they don't elongate. And then we've got another class that's dominated by neural cells, but these don't look like the typical neural cells. They look like they're hindbrain, and we also don't get elongation. And these are the ones that we don't see with conventional polyclonal gastruloids. Okay. So now, we can kind of rephrase our question a little bit. So we've got our monoclonal gastruloids, right? And we see a lot of heterogeneity. So now we're going to ask the question, when is the heterogeneity emerging, right? When in this process is it occurring? Because in each case, we're now starting with a single cell, right? Is the heterogeneity pre-encoded in that first cell, or is it kind of emerging along the way? And for this question, this is where we go to rely on DNA typewriter, right? And so here we're encoding the components I talked about before. We have lots of instances of tape integrated throughout the genome, now not of HEC293T, but of an embryonic stem cell. We have many PEG RNAs, and we have a DOX-inducible prime editor, okay? And we're putting these all into a clone. So we now have a monoclone that has all these components. And then we let that clone give rise to various stem cells, and these various stem cells become the founder cells of monoclonal gastruloids. And we turn on the typewriter as we are in the process of founding these monoclonal gastruloids, okay? All right, so this is kind of representative data, similar to what I showed before, but now we've got we not only know, every set here of the 8 or 10 sets shown corresponds to an individual tape. You can see that we're writing left to right. This is all monkey at a typewriter, and the colors correspond to different keys on the typewriter. And you can see the accumulation of information in a way that is coherent when we build a phylogenetic tree and tells us something about the cell lineage relationships. But now we also know which well each cell came from based on debris-seq, so you could think about debris. And we also know the cell types based on single-cell RNA-seq. And so we can assign these, and we can see the phylogenetic tree that we build is coherent in the sense that this gastruloid and this gastruloid are well separated in the tree. So that gives us some reassurance. Okay, so now this is now an example of two gastruloids that are, related to this. This come from this clone, and the resulting tree. And so again, the nice thing is, we see this strong separation of cells that were assigned to the wells corresponding to the two different gastruloids, which is reassuring. This is all done with like the other advantage of getting rid of the deletion issue that we had before is that we can rely on more conventional tools from the phylogenetics field, like bootstrapping. And we can show that, these are robust trees, particularly in the deep clades. And so we've got confidence in the tree itself. And you can see quite a lot of structure here on the outer ring, right, in terms of how cell types are grouped with one another or not in some cases, right? And we can delve into that a little further. This has been, I should say that the transition to this work was really led by, the experimental work was led by Sam and also Choi again. And then Chengsheng Q has been helping to drive the analysis. So, if we dig into some of these examples, some of this structure in more detail, I'll show you some cool examples here. So here's one where, now we're looking at this particular wedge here. So just this part, and we're looking at particular cell divisions, right? So here in this wedge, we have an inferred cell division, which occurred, just a handful of generations after the seeding of that stem cell to that well, the founding of the gastruloid. And we see this extraordinarily strong difference between the fate outcomes for the daughters of that cell division. One daughter will go on to give rise to, all kinds of cell types, but really to the parts of the gastruloid proper. I think the pinkish here corresponds to somites, right? So this is like somitic cell types. But the other daughter is entirely extraembryonic endoderm, right? So this is an example of early fate restriction, where probably one of those stem cells just differentiated spontaneously during the formation of the aggregate. There are other examples that I would characterize more as fate bias, right? So here we don't see such a strict restriction, but again, we have an early cell division where one daughter is overwhelmingly giving rise to mesodermal lineage and the other daughter overwhelmingly to neural lineages, right? Here's another one where this is now the first cell division after the founding of the gastruloid. So you have one cell seeded to the well, it divides, and one of those daughters is almost exclusively going to stay stem cell-like, right? Like it's not really going to differentiate. Whereas to the extent that there is differentiation to what we would expect in a gastruloid in this particular individual, that's all coming from the other, not all, but largely from the other daughter cell, right? So again, an example of very, not fate restriction, but very strong bias, right? And so the interesting thing is that, we have these and many other clear examples of these biases occurring within a handful of cell divisions of the founding of the gastruloid, right? And so the implication here is that there are fluctuations in the states of those stem cells occurring relatively early on that are either heritable or otherwise are somehow captured, whether it's in spatial structure or something like that, in a way that they become very predictive of what all of the daughters of that subclade will go on to give rise to that one branch of the early cell division, right? Okay. So now this is telling us something about the heterogeneity and where it arises in the context of a gastruloid. So within that gastruloid or intragastruloid heterogeneity. So we try to do one more experiment. I know I'm getting short on time here, so I'm going to go through this pretty quickly to leave some time for questions. But it's a pretty fun one here where we did what we called a tree of tree experiments, or a two epoch kind of thing, right? So In this experiment, we started with a single founding stem cell. We expanded that stem cell to many stem cells, recording this lineage this whole time. And then we took these stem cells and used them to found individual monoclonal gastruloids, right? So we have epoch 1 and epoch 2. Typewriter is going this whole time. And so now we can ask the question of whether the heterogeneity that must emerge during epoch 2, because that's when the gastruloid was founded by a single cell, is informed at all by the lineage relationships between these founders during epoch 1, right? And so this is a tree of roughly 100 monoclonal gastruloids. This is showing the epoch 1 lineage relationships of the founders, right? So what are the lineage relationships of the founders to one another? And then we're looking at whether we're seeing heterogeneity that emerges in the epoch 2 and whether that heterogeneity is informed by epoch 1. And to kind of cut to the chase, the answer is yes, it is informed, right? And so one of the challenges here is that this was the experiment in which we saw the least heterogeneity of any gastruloid experiment we've done to date. And we think that may be in part because now we've really made the founding stem cells as related as possible, right? By constraining the number of cell divisions between that founder of founders and the founders of gastruloids. But if we do capture the variation in various ways, and in particular looking at the proportion of cells in a gastruloid that will remain stem cell-like, and the proportion of cells in a gastruloid that will become hindbrain-like, we do see that there are subsets of gastruloids that are related to one another in this first epoch and do appear to have, more than expected by chance similarity with respect to these aspects of phenotype, which are the primary among the primary axes of variation. So again, I'm glossing over this for reasons of time, but I want to show one more slide here, which is kind of the lineage tree here. So here at the center, we have our single founding stem cell, and then the A, B, C, D, E, F correspond to major clades of gastruloids, and then the colors in the middle ring correspond to the individual gastruloids, and the colors in the outer ring correspond to the cell types. But now we have a tree of roughly 50,000 cells, all starting from a single stem cell that are, it's a tree of trees effectively. But it also, I think, is a nice proof of concept that the technology that we developed in HEC293 T cells can work in stem cells, and I think can scale. And so now we're hoping to take this to the next level. We have a new initiative in Seattle called the Seattle Hub for Synthetic Biology. And I had a few slides to tell you a little more about where we're headed with this, but I'm going to skip over those for reasons of time. And the primary message here is that, I think there's still a lot of work to do to get this, probably years of work to do to get this really to where we want it to be. But at least the path is looking pretty decent for getting there. And in the future, we hope to be able to record, again, much more than lineage, much more than enhancer activity, but there's a lot of work between here and there. And thank you for listening. And I try to acknowledge folks along the way, but this has been a lot of collaboration between labs with past and former trainees, and increasingly with entities like the Allen Institute. And thanks for listening, and happy to take any questions.